The cell cycle consists of interphase and mitosis and is necessary for sustaining life. Interphase, which includes G1 or cell growth, S or DNA synthesis, and G2 in which it prepares to divide, is the phase that cells spend most of their time in. Mitosis is the process by which a eukaryotic cell separates its chromosomes into two identical sets. It is primarily carried out in ordinary body or somatic cells. It is an important process in cell replication and organism growth. The daughter cells produced are diploid, represented by 2N, meaning that they each contain two sets of genetic information. In humans, a diploid cell contains 46 chromosomes. The first phase of mitosis is known as the prophase. During this phase, chromatin, genetic material located in the nucleus, condenses into what is known as a chromosome. In this stage, the nucleoli disappear and the mitotic spindle, the network of fibers that will eventually separate the chromosomes, begins to form. At this point, each duplicated chromosome is made of two identical sister chromatids joined at the center by their centromeres. Cohesin proteins hold the rest of the sister chromatid together. Slowly, the centrosomes will start to move apart, spreading out and developing the spindle fibers. The next phase of mitosis is the prometaphase. In this phase, the nuclear envelope begins to break down. The microtubules that make up the spindle are now attached to the now even denser chromosomes. One fiber connects to each side of the chromosome on a special protein known as a kinetochore. Metaphase is the next and longest phase of mitosis. In this stage, the chromosomes line up along the metaphase plate in the center of the cell. The centrosomes are at complete opposite ends of the cell and the spindle has completely formed. The spindle fibers are still connected to the chromosomes at their kinetochores. The next and shortest phase of mitosis is anaphase. To begin anaphase, the cohesin proteins are cleaved. This separates the sister chromatids into separate chromosomes. The two separate chromosomes begin moving to opposite sides of the cell towards the centrosomes, pulled by the spindle fibers. At this point, we see the cell shape begin to elongate as it prepares to divide. Telophase and cytokinesis are the final phases of mitotic division. During the telophase, the developing daughter cells begin to reform their nuclear envelopes and the chromosomes loosen in the nucleus. Now the original plan of dividing one nucleus into two identical nuclei is complete. Cytokinesis, or splitting of the cytoplasm, is the last step. A cleavage furrow, which pinches the cell in two, forms so the two daughter cells can separate. The end of cytokinesis ultimately leads to the beginning of the G1 phase, restarting the cell cycle.
Meiosis is important in sexual reproduction. Meiosis ensures that the number of chromosomes remain constant from generation to generation and that genetic recombination occurs. Genetic recombination ensures that the offspring are genetically unique from their parent. Meiosis has two phases, Meiosis 1 and Meiosis 2. Meiosis 1 separates homologous chromosomes and Meiosis 2 separates sister chromatids into 4 haploid daughter cells. Prior to meiosis, each chromosome is replicated so that the chromosome consists of two chromatids attached at the centromere. Sister chromatid are identical copies of one chromosome, but homologous pairs are two different individual chromosomes inherited from different parents. Meiosis 1 starts with prophase 1, where chromosomes condense and homologs pair together. Homologs undergo crossing over, which is the exchange of DNA segments between the homologous pairs of chromosomes. This occurs in the synapses, where the pair is held tightly together by proteins. During the middle of prophase, synapsis ends and the homologous pair begins to drift apart but remain connected by casematas, which are points where crossing over occurred and sister chromatid cohesion has formed. During prophase, centrosomes begin moving to the poles of the cells and spindle forms while the nuclear envelope breaks. Near the end of prophase, microtubes from the pole attach to the kinetochores of each chromosome and homologous pair. In metaphase 1, homologous chromosomes have moved onto the metaphase plate. All kinetochores from one chromosome from each homologous pair is attached to spindle from the one pole. In anaphase 1, homologous chromosomes separate while the sister chromatids remain together. This happens because cohesion proteins break down, so there is no casemata connecting homologs. The homologs move toward their pole on the spindle. The next phase is telophase 1 and cytokinesis. In telophase 1, each half of the cell has a haploid set of replicated chromosomes. All these chromosomes are made up of two chromatids. Cytokinesis begins to divide the two halves of the cell into two separate cells. In animal cells, a cleavage furrow forms, and in a plant cell, a cell plate forms to divide the cell into two halves. No replication occurs prior to meiosis 2. Instead, meiosis 2 uses the two cells formed in meiosis 1. Meiosis 2 begins with prophase 2. A spindle apparatus forms and attaches to the kinetochores of each sister chromatid. The sister chromatid move towards the metaphase. In metaphase 2, the sister chromatid are positioned on the metaphase plate. In anaphase 2, cohesion proteins break down so the chromatids can move toward opposite poles. Meiosis 2 ends with telophase 2 and cytokinesis, which divides the daughter cells into separate cells. Meiotic division results in 4 daughter cells. All 4 have a haploid set of unreplicated chromosomes that are genetically distinct. They have half as many chromosomes as the parent cell.
In both mitosis and meiosis, DNA replication occurs in interphase prior to the beginning of each process. In mitosis, one cellular division occurs, while in meiosis there are two divisions. Crossing over only occurs during meiosis. The final outcome of mitosis is 2 diploid genetically identical cells, while meiosis produces 4 haploid genetically unique cells.